Welcome back. So now we're going to move forward to the second part of the lesson, which focuses on the control of microbial growth. This is a lot of students' favorite part of the lesson, whether you're going into the healthcare professional area or any other field. It's really, a lot of the terminology and things we discuss in this part of the lesson is really applicable to your everyday life. And like I've mentioned before, especially now that you have lived through a pandemic, a lot of this terminology and the concepts we talk about with regard to the control of microbial growth will be familiar with you and will be very relevant to practices you should kind of think about every day. Now, when we go through this section, please keep in mind that there's a lot of terminology. And so when you are going through these slides, make sure you know the definitions of each of these terms, especially the fact you'll see them bold, colorful. When you know the definitions, know what I call in the way backwards and forwards, meaning if I give you a term, you should be able to define it. But also, if I give you a definition, you should be able to identify what term I'm talking about. In addition to that, you'll notice that each slide will have a few different terms. You should be very comfortable with distinguishing or knowing the similarities or differences between words. So for instance, in this lesson, when we talk about disinfectants versus antiseptics versus sterilization, make sure you not only know the definitions of each, but kind of what distinguishes one from the other. Okay, so we're going to start heading into terminology now. Sepsis versus asepsis. Okay, sepsis is a term that you will hear a lot, unfortunately, in hospitals now. It is a big killer of a lot of patients, and what's actually killing them is you guys, the medical professionals. The medical professionals are spreading infections to their patients without realizing it when they're not thinking in terms of microbiology. Okay, so when you hear sepsis or septic, that means that there is bacteria present, okay, bacterial contamination. Whereas when you hear asepsis or aseptic, that means sterile, no bacteria. Okay, so you want aseptic conditions. You want asepsis. Then we have three terms that I'm sure you hear all of the time, but still so many times students don't actually know what they mean or how to distinguish them. Okay, so I want you to put a star next to this slide and make sure you know the difference between disinfection and antisepsis. Okay, meaning the difference between using a disinfectant versus using an antiseptic. Okay, this is important because I never want to see you trying to put bleach on one of your patient's skin. Okay, because bleach is a disinfectant, whereas only antiseptics get used on patient's skin. Okay, so disinfectant or disinfections, these are things that you use to kill microbes on inanimate objects. So for instance, cleaning a table, cleaning desks or pens or pencils, anything like that, that's disinfection. Okay, using Clorox, Lysol, that's disinfectants. Whereas antiseptics, what you can do is you can underline TIS in the word antisepsis or antiseptics. That tells you that you're using these on living tissues. Okay, TIS, just like tissues, living tissues and skin. So for instance, when you use Purell, that's an antiseptic. When you use iodine on your patient or alcohol, okay? Now another term that's related to these but also different is sterilization. And what sterilization means is you are getting rid of 100% of the microbes. Now, why is sterilization considered different or separate from disinfection and antisepsis? Well, think about what you see on your Purell or your Lysol and Clorox. What does it always say? Kills what? 99.9%. Okay, it does not kill 100%. If you want sterilization, then you'll have to look for some other means. So for instance, things like autoclaving or gaseous agents, but these are things that cannot be used on patients, okay? So a lot of times sterilization has the downside that it would be too dangerous for patients. Now some more terminology we have here. We have first up degerming, okay? Notice degerming is removal of microbes by physical means. You are not killing them. You're just physically wiping them off or washing them off. So the picture on here, degerming, would be washing your hands, for instance, okay? Because you're using water and you're using the movement of your hands to brush off microbes. Sanitization, that's when you disinfect someplace used by the public, okay? So if you were to clean the desks in the school or go to the court cafe and wash those desks, wash those tables, or even for instance in a restaurant like McDonald's or something, you're wiping down, you're disinfecting those tables. That's sanitization, okay? Whereas if you're just wiping down, cleaning your own personal kitchen table that's not used by the public, then that's just disinfecting, okay? So sanitization or sanitizing is specific to something used in the public. Then we have pasteurization, and you ask yourself, where do you hear this term? Milk, right? Milk is pasteurized. So what they do is they heat to kill the pathogens, but they're not sterilizing. They're not killing 100%. And if you think about our example of milk, if you try to heat it to a temperature that would sterilize, well, you would curdle the milk. You would make the milk no longer good. Okay, so it's kind of, they want to heat it to the point that you get rid of a lot of the pathogens, but they can't completely sterilize it because that would destroy the product. And then we have two terms that you have seen before that we've mentioned earlier, which is bactericidal versus bacteriostatic, and you have to know the difference between these. Okay, these terms will keep coming up throughout the semester. So remember, cidal kills, static slows or stops. And always ask yourself in terms of temperature, what's bactericidal? Very hot, right? Things like fire, flames, cooking. What's bacteriostatic? That would be cold temperatures, putting something in the fridge, in the freezer, okay? Then we have here various factors that can affect the rate of microbial death, okay, from antimicrobials. Now that's a fancy way of saying for each of these factors, you have to ask yourself, okay, does this factor make it more likely that microbes will be killed? Does it make it easier to kill the microbes? Or does it make it harder where less microbes will be killed? Okay, so you're increasing the rate of microbial death if it's something that makes it easier to kill microbes. Okay, the first category that we have is the number of organisms. Okay, the number of organisms, think about it, if there are more microbes present, more bacteria, is it going to be easier or harder to kill them? Okay, it'll be harder to kill them, right? It'll take a longer time. So more microbes decreases the rate of microbial death from antimicrobials. It makes it harder to kill them. The second thing we have is duration of treatment, okay? The longer you apply a disinfectant or an antiseptic to something to try and kill bacteria, the more of it you will kill, okay? This duration aspect is why alcohol is not a great antimicrobial, because think of it, what does alcohol do very quickly? It evaporates. So that's short duration of treatment. Okay? If you're not going to have that chemical on the surface for very long, it's not going to kill as much bacteria. Okay? So longer increases the rate, whereas shorter decreases the rate of microbial death. Okay? Then we have temperature. And if you think about it, lower temperatures like the cold take longer to kill microbes. It preserves them. Whereas hotter temperatures, what do hotter temperatures do? They're bactericidal, right? So they kill better, okay? They help you kill the microbes quicker. Then we have the next one, which is my favorite. That's the environment. But when we talk about the environment, we don't mean trees and greenery and all of the environmental stuff like that. What we mean here is the presence of organic material. All the fun stuff that nurses love dealing with, like vomit, blood, pus, saliva, urine, any of that, okay? These organic materials, they decrease or drop the rate of microbial death by antimicrobials for a couple of reasons, okay? If you picture these, I know you don't want to do this, but picture a whole bunch of vomit right on your patient's lap, right? And you go to clean it up. Well, microbes can be hiding in all of those little crevices, okay? It gives them protection. It makes them less accessible to you, okay? So it protects them and decreases the rate of death. It also helps destroy some of the chemicals that you will try to use to kill them, okay? So certain things like vomit, sometimes the pH of it or various aspects of it can kind of help break down disinfectants and make them less effective. Okay, so organics are bad for trying to kill microbes. Then the last category we have are endospore formation. And for that, you just have to think of what exactly is an endospore. Okay, and we mentioned those are basically those nice protective little escape pods. They're very resistant. So if they're very resistant, then what does that tell you about you trying to kill them with antimicrobials? It's going to make your job a lot harder, isn't it? Okay, so endospore formation will decrease the rate of microbial death. It will make it harder for you to kill the bacterial infection if they are capable of making endospores. Okay, so again, for any of these factors, you have to ask yourself, will it make it easier or harder for me to kill the microbe? Then we think about what are some actual examples of antimicrobials and how are they working? Okay, so you don't have to memorize all of these, but I will have you star certain ones to focus on. Okay, the first one we mentioned are phenols. These are very rough, harsh agents. They tend to destroy bacteria by messing up their plasma membranes and by denaturing their proteins. Then we have alcohol. And I want you to put a star next to that, put a star next to it, and ask yourself, why is this one not a great antimicrobial? Well, we just mentioned it a minute ago. It evaporates too quickly. Okay, it evaporates too quickly. So whenever you see alcohol, that's what I want you to remember. Then we have iodine and chlorine. Okay, so iodine you've probably encountered in the hospital already. Whenever you see the word tincture, that simply means that it's been mixed with alcohol. Okay, certain things they mix with alcohol to kind of combine the antimicrobial activities or because it doesn't work as well without that solution. Then you have surfactants, and notice I put two of these in red. Put a little star next to these guys. I want you to remember the difference between soaps and detergents. Okay, two terms you hear all of the time, and people tend to use them interchangeably, but they're very different in micro. Okay, there's a very big distinction between them. And what that is that soaps simply make things slippery, okay, which means they're good at degerming, because remember, we said degerming means that you wash away, you physically brush off bacteria, get rid of it. But what are you not doing to it? You're not killing it, which means that it's a poor antimicrobial. Whereas detergents kill the microbes. Detergents, they disrupt or destroy the membrane, the outer part of the bacterial cell, which means they are great antimicrobials. They kill the bacteria. Okay, so remember, slippery soaps are not good antimicrobials. They're not killing, whereas detergents are killing bacteria. Okay. The last one I want you to remember is gaseous agents. Put a little star next to that one because what do you notice? All capital letters. Gaseous agents sterilize. Okay? They sterilize because they could get into every little crevice, every little pore of that microbe. Okay? The problem with them, though, is they're usually too harsh to be used anywhere where a human would be. Okay? So you have to think about, yes, I want an antimicrobial that will kill as much bacteria as possible, but I also don't want to hurt the humans that are around there. Okay? So it's a tricky little juggling act that you have to do. Now, when you want to see how well a chemical agent works, we use the disk diffusion method, which you guys will actually be doing in the lab, okay? And one of the examples of this will be Kirby Bauer in the lab, but you could do it with any disinfectant or antibiotics as well. Basically, what you do is you swab one of the agar plates that we use in lab. You completely swab it with the bacteria of your choice. So for instance, here we have Staph aureus, E. coli, Pseudomonas. So you completely swab it, and then you place these small white filter discs on top of it. And these filter discs you first soak with the disinfectant. You then incubate the plate and you see, where does the bacteria grow? Where does it not grow? So you'll notice that for some of these, like around chlorine, you have this big circle, this big zone of inhibition where no bacteria grew because that disinfectant killed the bacteria. It's very good at killing. Okay? That big zone means that that bacteria is very sensitive or susceptible to that disinfectant. Okay, so you would want to pick that disinfectant to kill it. Whereas you'll notice Pseudomonas all the way on the right, chlorine works a little bit, but it's not as susceptible. Instead, it's rather resistant. Okay, any of the ones where you see the disc with no empty halo around it, no zone of inhibition, that means that that bacteria is resistant to those disinfectants. Okay, so for instance, you would not want to select hexachlorophene to try and kill Pseudomonas or E. coli, because notice that the bacteria grew right up against the disc, no problem. Okay, they were resistant to it. The last thing that I want to mention is some physical methods to control microbial growth. Okay? Physical methods include heat, filtration, temperature, high pressure, desiccation, osmotic pressure, and radiation. Okay? So when we talk about heat, you notice there are two forms. There's moisture heat and there's dry heat. So in the lab, we use a lot of times the incinerators on your desks. That is dry heat. Or if we use a Bunsen burner, that's dry heat. And those sterilize. Okay? They completely kill bacteria. We also use the autoclave, which is this giant metal contraption down the hall, that also sterilizes. And that one sterilizes by using high pressure and water and steam, very high temperature. There's also filtration, which we'll talk about in a minute, and why we use heat versus filtration. Then there are low temperatures, and for low temperatures, notice we said we're doing microbial control. We did not necessarily say we're trying to kill the bacteria. Okay, so low temperatures, unlike heat, low temperatures will just slow the growth. So just remember that when you deal with putting food in the refrigerator and the freezer, you can't leave it there forever. Okay? In the fridge, you only want to leave it for a few days. In the freezer, maybe a few months, and then toss it out because the bacteria does still grow. It's just at a very, very slow rate. Okay? High pressure, that's important as a microbial control because that can denature proteins. So that's why the autoclave not only uses high temperature, temperature, but it also uses high pressure, okay, because denaturing proteins, that kills bacteria. Okay, so it's bactericidal. Then we have desiccation, and desiccation is a fancy way of saying completely drying out. Okay, so if you shrivel the cells, if you get rid of their water, you've now killed them. That prevents metabolism, that prevents any of their reactions from going on. Okay. You also have osmotic pressure, which we've mentioned before. Osmotic pressure is using salts. So think about when they used to preserve foods by having lots of salt, okay, like preserving meats with tons of salt, because that is a hypertonic condition, okay, so it shrivels up the cells and they cannot perform their life functions. The last one here is radiation, and radiation, as many people tend to know already, damages DNA, okay, so it's not just a problem for the bacteria, but it's also dangerous for us as well. Okay, so there are just a couple of these I want to give you a little more information on before we finish up. The first one here, we have autoclaves, and in giant letters on here, I want you to write sterilizes. Okay, autoclaves sterilize. They kill 100% of the microbes, including any endospores. So that means any of those highly resistant cells. And they do this by using very high temperatures and extreme pressure, which we just mentioned a minute ago, that denatures proteins. Okay? And in the figure here, you see the two different colors of the tape. You have the yellow and the blue tape. That's basically just so that the person autoclaving knows that that reached the proper temperature and pressure to be sterilized. Okay? But for autoclaves, I want you to know high heat, high pressure, sterilizes. Now, we also have filters as an option. I want you to know the difference between using an autoclave and using a filter. So we said autoclaves are great in that they sterilize 100%. But ask yourself, when can't you use an autoclave? Well, certain materials, if you were to put them in high temperature, high pressure, certain containers might completely melt, collapse, break. Certain reagents, what can happen if you put them in high temperatures? Boom, they might be flammable. Okay, you might cause an explosion or a fire. Okay, you can destroy what you're working on with an autoclave in some certain scenarios. Okay, so filters you use when you cannot use high heat. Okay, so when would you want to use a filter? When you can't use high heat. When the heat would destroy your container, your sample, or when things are flammable. Okay, so instead you put them through a filter. Now notice, what is a filter doing? It's separating things out, right? What's it not doing? It is not killing the bacteria. It's just removing them. Okay, so keep in mind the difference in this. And filter size matters. So depending on the pore size will determine how much microbes you get rid of. Okay, and you'll notice HEPA filters, those are the ones you hear with your air conditioning units in your houses. Okay, they will have bigger pores, whereas membrane filters are more expensive. Those are the ones we use in research. Those have much smaller pores to really cut down on the microbes. The last thing to mention is radiation. So this is in things like microwaving your food. Okay. Now with radiation, as we've mentioned before, and you've probably heard previously throughout your life, radiation is dangerous in that it breaks DNA. So it not only breaks the DNA of the bacteria, but it can also break your own DNA. So always be careful with the idea of radiation as an antimicrobial. Now with the way microwaves work, they're not just using radiation in the sense of trying to break DNA, they're producing heat. And as you know, what does heat do? Heat kills microbes. It is bactericidal. Okay? So you want to just remind yourself in terms of radiation or in terms of any of the antimicrobial techniques we talked about, how is it good at killing microbes, but why can't we use it under certain conditions? Okay, what's the downsides or limitations with it? Okay. Okay, so now we're going to switch over to the very last part of this lesson, where we're going to focus on metabolic diversity and functional diversity. And we're only going to cover a few of the concepts or a few of the terms, because honestly, those chapters I don't find very relevant. They're highly memorization based and things that you don't actually encounter or utilize on an everyday basis when you're in the world of microbiology or when you are in the medical field. Now, the first thing I want to point out is when we say metabolic diversity, well, what exactly do we mean? So when we say metabolic diversity, we define it as different metabolic strategies that organisms have evolved to obtain energy. And as you have kind of seen through the previous lecture as well, when we talk about metabolic or metabolism, we're talking about the chemical reactions within an organism. So what are these organisms capable of doing in terms of their chemical reactions? How have they kind of evolved or come up with different ways to break things down or to obtain energy or things like carbon, as well as other requirements that we just talked about in the earlier slides. Now, when we talk about things like metabolic diversity, it's also always important, in addition to a definition, to kind of know what are some examples, because that helps you understand what exactly we mean. So when we talk about metabolic diversity, we're going to talk about phototrophic mechanisms, autotrophic, chemolithotrophic mechanisms, nitrogen fixation, sulfur, and iron oxidation. Now, I know right now all of that sounds like gibberish or another language, but in the coming slides, we're going to define and kind of talk about what each of those things mean. So make sure that by the end of this lesson, you kind of know or understand what each of the terms I just mentioned were. Know what phototrophic is referring to, or autotrophic, chemolithotrophic, things like that, okay? Now, the first one we're going to cover is phototrophy. This one's usually rather easy for students to figure out what it's talking about, because when you hear photo, you think about light, usually in the sciences. So phototrophy, or being phototrophic, means the ability to carry out photosynthesis. And you all have probably heard that term before. Photosynthesis simply means that they are using light energy. They're going to use that light energy in order to make what's necessary for those cells. Now, when we talk about phototrophy and the process of photosynthesis, I want you to know that photosynthetic organisms also are what's called autotrophs. And autotrophs, we've mentioned that term before in the previous lesson, that means these organisms can use carbon dioxide as their main carbon source. And when you think about it, well, carbon source, what do cells need carbon for? In order to make all of the biomolecules. Your cells can't make things like proteins, carbohydrates, lipids, nucleic acids, if they don't have usable carbon. So autotrophs are organisms that are able to use CO2, carbon dioxide, as their main carbon source to make their biomolecules. You, on the other hand, are not an autotroph. You are a heterotroph. Okay, that means you have to take in or eat carbon sources. So you have to, for instance, eat sugars and proteins and carbohydrates. You have to have meals in order to isolate the carbon from those food products, from those nutrients. Now, going back to phototrophic and the idea of photosynthesis. If an organism is going to use photosynthesis, meaning use light energy, what they end up having to do is two types of reactions. They have light reactions and they have dark reactions, also known as light independent reactions, because they don't require light. Okay, they're independent of light. The purpose of light reactions in photosynthesis is simply to produce ATP, and you can see that in this figure here. Over here, notice that this side of the figure represents the light reactions occurring thanks to this photosystem set up over here. And notice that these light reactions are producing ATP, okay, energy. The dark reactions, on the other hand, light independent, that's this right half of the diagram. And what do you notice is being made here? Sugars, right? Valuable material. And what are they using? Carbon dioxide. So when we mention dark reactions, I want you to write down that dark reactions are when the organism will reduce carbon dioxide, which you see over here, reduce it into usable material for their growth, specifically in the form of sugar, that they'll then use for things like cellular respiration or for making their various biomolecules. Okay? Now, we're not going to go into the details of exactly how they're doing this in terms of the pathways because this isn't biochemistry. But I do want to point out that when you hear the terms photosystem 1 and 2, that right here is associated with the light reaction, whereas the Calvin cycle over here is focusing on part of the dark reaction. Okay, but I just simply want you to know when you hear light reactions, what's the cell doing? Producing ATP. When you hear dark reactions, what's the cell doing? It's reducing carbon dioxide to usable material, specifically sugars. Okay, that was a whole lot of information, and sometimes YouTube kind of messes up closed captioning or whatnot. So I have typed out a transcript of what I just said in the previous slide, so you can pause, jot down your notes, and then continue on. Now, when we talk about phototrophy, I also want you to know the difference between oxygenic phototrophs and anoxygenic phototrophs, okay? When you see oxygen in the word, well, that kind of gives you a hint as to what's being involved here. So oxygenic photosynthetic organisms, or phototrophs, what they do is they release oxygen as a waste product. And so you can see that in this half here. So oxygenic phototrophs release the waste product of oxygen. Whereas when you see anoxic or anoxygenic, that's telling you no oxygen. So anoxygenic organisms do not produce oxygen. And notice on the anoxygenic side, you don't see any oxygen produced. Now, when we talk about each of these organisms, I want you to know some examples of what we're talking about. So when we say oxygenic, I want you to circle, star, highlight cyanobacteria, this word right here. Okay, you find those especially out in the waters, out in the oceans, and in fresh water as well. And so the example, oxygenic, think cyanobacteria. Whereas anoxygenic, those are going to be all other phototrophic bacteria, which includes purple and green sulfur bacteria. And you're going to see those terms again in a couple of slides. Now, the reason why distinguishing these is so important is because oxygenic phototrophs, specifically or especially the cyanobacteria, they're responsible for what's called the Great Oxygenation Event that happened billions of years ago. And basically what they did is they converted Earth from being anoxic, meaning not having an atmosphere with usable oxygen, and through their phototrophic activities, they released the waste product, which to them is just waste, but to us is very valuable. They released the waste product of atmospheric oxygen. So they gave our atmosphere the oxygen, the form that we need in order to survive. Now, of course, they did this billions of years ago before we were on the planet, but ultimately we would not have ever been able to exist here if it were not for the oxygenic cyanobacteria back in the day giving us that waste product of oxygen in the atmosphere. So oxygenic versus anoxygenic are very important. And like I said, especially that example of cyanobacteria, because that gave us the great oxygenation event. Again, since that was a lot of information and some terminology that you might not know how to spell, I made sure to type out a transcript of exactly what I was saying so you can pause and jot down your notes. Okay, so the last terms we're going to talk about with regard to phototrophy, again, the idea of photosynthetic organisms using light. The last two terms are carotenoids and phycobiliproteins, as well as phycobilisomes. Okay, so first up we have carotenoids, and what those are basically beautiful pigments actually. They're yellow, red, brown, green pigments that are embedded in photosynthetic membranes, okay, the parts of the cell that's going to be responsible for photosynthesis. And their purpose is not only to help absorb a little bit of energy, but they also serve as what's called photoprotective agents, meaning that carotenoids, they absorb, or you sometimes hear it as keratin, they absorb the light that would be harmful or toxic to cells. And what they end up doing is they protect those cells from photo damage by blocking that particular light that usually would trigger chemical reactions producing toxic forms of oxygen. So instead, by blocking that particular toxic light, they're allowing for good processes rather than the bad chemical reactions. And that's why you'll notice they're used in things like looking at human health and age-related issues, okay, because they're basically protective pigments. You also have phycobiliproteins and phycobilisomes. Sometimes you see phycobiliproteins written out as PBPs, so that right here in the figure, that's PBPs, that's phycobiliproteins. Okay, fun word to say. These are pigments in cyanobacteria and red algae that basically serve as the light harvesting system for phototrophs, meaning they're going to be responsible, these pigments, for capturing light that then goes through the photosynthetic reactions that we've mentioned already. So what then happens is these PBPs, the phycobiliproteins, they basically assemble into these bigger clusters called phycobilisomes. Okay, so the phycobilisomes are just clusters of the phycobiliproteins, and that phycobilisome structure ultimately attaches to the thylakoids, and thylakoids are the membrane-bound sacs or compartments where light reactions of photosynthesis occur in photosynthetic organisms, whether it's a microbe like cyanobacteria or whether it's plant cell. Now, what's a fun fact is if you look at these figures, you see that cyanobacterial phycobiliproteins as well as carotenoids, they have commercial uses. A lot of things that you use on a daily basis, especially if they're beautiful colors, for instance, some of the natural cosmetics, they use these pigments to help give, these natural beautiful colors to things. Okay, so when you see carotenoids and phycobiliproteins, automatically think pigments. Okay, the carotenoids being more yellow, red, brown, kind of the things you would see in autumn, the leaves, those pigments. Whereas the phycobiliproteins, those a lot of times you see very vibrant kind of colors as you see in the picture here. Okay. Again, that was a whole lot of words, so I typed out exactly what I was saying to kind of be my own closed captioner, since sometimes YouTube messes it up a little bit for the scientific terms. Okay. Again, when we go through this kind of terminology, feel free to contact me in the Remind app at any point if you need any terms better explained or any other examples, or if you miss any of the answers to questions that I have on the slides. Okay, never hesitate to contact me in the Remind app. So as I said before, phototrophic organisms, the organisms using photosynthesis, they're also autotrophic. And what autotrophy means, we've mentioned it before, it's when an organism reduces carbon dioxide in order to use for cell material. So it's basically using carbon dioxide as the main or major source of carbon to then make its biomolecules, for instance. Because remember, carbon is for all biomolecules, meaning proteins, lipids, carbohydrates, and nucleic acid. All four of those need carbon in order to build them or make them. And in order to do this, in order to reduce the carbon dioxide to make cell material, they use the Calvin cycle, which you saw in the previous slides, okay, in the dark reactions of photosynthesis, the light independent reactions. They were reducing carbon dioxide to make sugars and specifically then carbon for the cell. Now, in addition to the Calvin cycle, they also sometimes use what's called the reverse Calvin cycle or the reverse citric acid cycle, as well as some of the other pathways that I wrote on the slide. But again, we're not going to go into the details of any of these pathways because that's more of a biochem thing or memorization thing. I just want you to be aware of what autotrophy actually means. Now, in addition to autotrophy, another aspect of microbial metabolic diversity is nitrogen fixation, and that's really important because basically, if you think about it, cells don't only need carbon, they also need nitrogen. The trick that I use to remember why they need nitrogen is I think of NAN, kind of like Nana. So NAN, I think nitrogen is for amino acids and nucleic acids, okay? N for nitrogen, A for amino acid, N for nucleic acid. So that basically tells you that cells need nitrogen in order to make their proteins and to make their nucleic acids, which are mainly DNA and RNA. Sometimes you also see ATP categorized in there as well, but mainly focus on proteins, DNA, and RNA. Now, in order to do this, to do nitrogen fixation, they use an enzyme called nitrogenase, and they end up reducing N2 into NH3. Okay, but again, don't focus so much on the pathways, more the idea that they're using an enzyme called nitrogenase in order to make nitrogen available for the cells. There are also sulfur oxidizing and iron oxidizing bacteria as well. Here's the transcript or closed captionings of what I just said for you to jot down. So the first one, sulfur oxidizing bacteria, although it's great that they're capable of utilizing sulfur and breaking down, the problem with sulfur oxidizing bacteria, or what we call sulfur chemolithotrophy, meaning they are getting energy from inorganic compounds, specifically in this case sulfur. The problem with this sulfur oxidization process is that it results in a very acidic environment. Okay, so the byproduct of sulfur chemolithotrophy or sulfur oxidization, so same thing, just saying chemolithotrophy so that you remember that that means the organism is getting energy from an inorganic compound, in this case sulfur. The problem with this is that you get the acidification of the environment. Now that's problematic for things like gas pipelines because they end up getting destroyed by that acid byproduct, the change in acidity when those kinds of bacteria are around. And the main bacteria that does that, the main sulfur oxidizing bacteria that you'll hear about is called Theobacillus. And as soon as you hear bacillus, that tells you that it is a rod-shaped bacteria. There are also bacteria that can oxidize iron. Okay, so they are iron chemolithotrophic, meaning they get energy from breaking down inorganic iron. Now, this reaction unfortunately only yields a small amount of energy for them, so they end up needing to use a whole lot of iron. And what's interesting about this is those bacteria, as a byproduct, they produce these insoluble ferric oxide molecules, which basically appear as this brown, gelatinous, rust-looking slime wherever they are. And that's why in the picture here on the slide, when you have iron oxidizing bacteria growing, you see this collection of orange slime. So in this serial dilution picture, you have barely any bacteria, and then in the media, you gradually see that orange because you have a whole lot of iron oxidizing bacteria in those last samples. Now, the best known examples of iron oxidizing bacteria, again, you can call it iron chemolithotrophic. They are Acidotheobacillus, again, bacillus meaning rod-shaped, and Leptospirillum. Okay, so that's a spiral-shaped bacteria. Now, when I say that these organisms are chemolithotrophic, again, make sure that you write down chemolithotrophic means that they are using inorganic compounds to get energy. That's as opposed to our cells, which are chemoorganotrophic, which use organic compounds, specifically glucose or sugar, to break down for energy. Now, I also mentioned the term acidophilic when I talk about the byproducts of iron oxidization. And acidophilic means that these organisms have the capability of surviving in very acidic, low pH environments. Okay, and again, I'm going to give you a recap slide to help you jot down all of the stuff that I just said. And here are the recap notes for you to pause and jot down. So the last thing I want to mention is the idea of functional diversity. And again, you have to first ask yourself, well, what exactly does that term mean? So functional diversity is diversity in form and function as it relates to microbial physiology and ecology. So it's basically looking at common functional traits among organisms and looking for these links in terms of biodiversity patterns and the ecosystem, basically how everything around that organism is functioning. It allows for determining trophic relationships and interactions, so things in terms of how are they getting their energy, what are they taking in, what are they breaking down, what are they capable of taking in, and what are they capable of breaking down. Kind of, that idea of the metabolic relationships that we just spoke about in the previous slides, as well as their participation in different types of environmental cycles and adaptations that kind of allow them to deal with their environment. All of that is kind of looked at in terms of functional diversity because it's looking at the form and function of microbes, particular traits. Now, some of the group examples that they like to look at are things like purple sulfur bacteria versus green sulfur bacteria. They look at the nitrogen fixers that we've talked about, all of the different categories of organisms that we just talked about. For instance, the ones that have the genes or the proteins and enzymes necessary to break down or to utilize that nitrogen, the ones that are capable of sulfur oxidizing, iron oxidizing organisms, all of that, in addition to being metabolic diversity. Basically, when you then look to see the traits, the proteins, the enzymes that let them do those things, then you're looking at functional diversity. And the way we visually represent that is by phylogenetic trees, which you can see here and over here. Basically, these are used a lot in terms of evolution and ecology, looking at the different species, different organisms, and how they're connected. You know, ones that are sharing branches in the phylogenetic tree will have more commonalities versus ones that are further away in the tree. And it helps you visually see, okay, which ones have, you know, which shared traits, which ones can oxidize sulfur, which ones can oxidize iron, things like that. Okay, so we use phylogenetic trees. Quick recap, even though there wasn't as much information on that previous slide, just to give you a chance to pause and jot down some notes. And that is it for this lecture. As always, if you have any questions, concerns, you need a little practice problems, a little extra practice or help, just feel free anytime, day or night, contact me in the Remind app with your questions. If there's any slide you need further explained, just send me a picture of the slide and kind of ask me, can you explain this a little more? What should I know for this slide? Things like that. Okay? As always, make sure you head over to the discussion board and also make sure that you're adding today's lecture information into your study guide. Okay, again, any questions about that as well, just send me a message. Thank you and have a great day.